Okay, I'm going to introduce myself now. I'm Mary Zielinski. I'm a professor in the Division of Reproductive and Developmental Sciences at the Oregon National Primate Research Center and also in the Department of OBGYN at Oregon Health Science University. This is not the typical talk I usually give at this meeting, and I hope you'll bear with me as I represent some recent discussions we've had on these very issues that David has raised. So we are facing some difficult roads ahead, but they will have beautiful destinations in helping our patients achieve their dreams and their goals of having children. So originally, we were going to have a representative from the FDA here to discuss things from their perspective. And I did find a contact, so we do have a contact there who specializes in reproductive devices. And I was interested in speaking with him from the standpoint of how can we develop and what do we need to do to develop vitrification media. And I'm not going to talk specifically about that today, but he was a great help. He cannot provide advice, but he can definitely point you in the right direction. And I had to send him very specific questions. Then he would answer what I wanted to learn about. So Dr. Hung is not with us today, but this is his contact information I have if anybody has further questions about the minutia of FDA regulations or particular websites that would be of help. Just let me know. So in 2019, we know that the ASRM released its practice committee report on fertility preservations for patients undergoing gonadotoxic therapy and declared that ovarian tissue banking is an acceptable fertility preservation technique and is no longer considered experimental. So we have to consider now, as David mentioned, the safety of collecting ovarian tissue, the methodology we use to cryopreserve the tissue, and then again, what it needs to be done to transplant the tissue. So this recommendation by the ASRM was made looking at a lot of the data that was saying that this was safe to do the surgery to collect the tissue. Cryopreservation didn't seem to affect the tissue, and they specifically looked at results when the tissue is transplanted orthotopically, but we know there are other sites it can go. They considered this critical advance for the practice of fertility preservation in cancer survivors and a great boon to the quality of life of patients who would love to have their own biological children. And this is the only method declared to preserve fertility for prepubertal girls, since ovarian stimulation and IVF are not options. And but we have to remember that the pediatric population is very underrepresented in the studies that were used to generate the ASRM guidelines, and there's only a few case reports of actually children born following transplantation of ovarian tissue cryopreserved when the mother was peripubertal or prepubertal. And I would like to refer you to these two beautiful references here by Raul Duncan and Monica Laranda and Lena Nahata's group that really drill down into all of the things we don't know about pre- and peripubertal tissue and what we need to consider moving forward. So it's considered standard practice, but now we have to face the elephant in the room, as David introduced. Our tools and technologies are not currently regulated in the US. So OTC continues and TTC under IRBs for some clinical practices, but not for others. Some maintain IRBs just for children, but no longer for adults. There are no approved media on the market, as David mentioned, for fertility applications, and the current Oncofertility Consortium slow freezing media is not FDA approved. So before we all panic, there are some things that we can do, but we need to be ready to do them immediately. The practice of medicine, I was told by Dr. Hung, is not under the jurisdiction of the FDA. But as David mentioned, there are other concerns regarding if your processes are not regulated, some bad things could happen in the future. So if we're using non-FDA approved products or practices and something goes wrong, there's no record of these adverse events on file. And that leaves things wide open for patients and their supporters to make some problems. So currently, purified water and mineral oil used in ART procedures are exempt from FDA approval, but media will never be exempt. And Dr. Hung told me there's no grandfathering in of any media, regardless of how many publications there are. It's considered a medical device and will eventually need approval. So Kyle reminded me of this wonderful reference that summarizes pretty much all of the slow freezing procedures that are used around the world. And I'm going to blow up this table a little bit, but I want to alert you to this beautiful reference that summarizes what everyone's been using around the world. And so I rearranged this table to show you the different cryopreservation media and lumped them together by country and who's using what particular media. So I'd really just like to draw your attention to the top two rows. The Oncofertility Consortium slow freezing medium is currently in a diluent that we call holding medium, that is a MOPS buffered diluent. And the actual cryoprotectants are ethylene glycol and sucrose. In each of these tables, and there's also a protocol available on the Oncofertility website for the actual slow freezing process. And we only have currently now one attempted transplant here in the United States. This media is pretty much identical to what is being used by the group in Denmark, Klaus Andersen's group. Also a MOPS-based buffered media. And they do have healthy children born from ovarian tissue cryopreserved with slow freezing using this particular media. And they have babies from both orthotopic and heterotopic transplants. And you can go down the list to see all the different formulations. So again, we have nothing standard around the world. But most of these cryoprotectants have yielded healthy children, the majority from orthotopic transplantation and a few from heterotopic. So we're hoping that some of the data that's already out there from non-US countries will give our FDA some confidence that these media will be safe to use. There are two commercial vitrification kits in the US. One is based on the methodology that Sherm Silber developed, and the other is from the Japanese group by Dr. Suzuki. And neither of these are FDA approved. They're out there to use, but haven't gone through any of the regulatory processes. Healthy children have been born from both of these vitrified media. So I'd just like to summarize a recent meeting that some of us had with Cooper Surgical, who is the current manufacturer of the OC holding medium serum protein supplement and the slow freezing medium that the consortium uses. So we are the people listed there on the bottom, David, myself, a number of people who are here today who are important stakeholders in this very issue. And we spoke with Eva Farbach-Graubal, who's the Senior Director of Consumables Product Management at Cooper, and David Morrell was there as the Director of the Clinical Cooper Surgical Fertility branch. And so I'm going to give you good news and challenging news. The good news is that Cooper Surgical will continue to make all of these OC products for the next 10 months. So we'll still have access to them for a while. But the challenging news is that Cooper Surgical has had an IRB with the OC for about 10 years. And their Regulatory Affairs Committee noticed that, oh, wow, this has been going on for 10 years. How come we don't have a product? And so should Cooper Surgical continue to provide this service? And that's what led to this discussion. So the really good news is that Eva and Cooper Surgical are very committed to pursuing the necessary regulatory pathways to get FDA approval for this media. But there's a lot of work to be done. We need to determine the best approach to take to transition these to commercial products. And we can consider OTC and the medias as a standard treatment for patients moving forward, or should we maybe perhaps to speed up the approval process, have this designated as humanitarian use device. And this might allow more flexibility in getting this off the ground faster. And we can make a case. Monica gave Eva some wonderful data to use for specifically for pediatric patients to help us define what might be feasible. But the definition of human use device is one that will treat or diagnose a disease or condition that is manifested in less than 4,000 individuals per year in the US. So we currently are under this number, but moving ahead in the future, will we have more? And what will we need to do for the long term? The challenging news is that the approach to take will dictate the data that's needed by the FDA. So do we do a standard treatment or humanitarian use? We need the data to support the application. And we'll keep pounding this home about data, data, data. And we need the data to support the safety as well as the efficacy of the media. So our unknowns currently are what kind of data does the FDA need? And that's what Eva is helping us trying to find out in her further discussions. We don't know the number of cases and associated patient data, even just within our own consortium here. So in having this meeting with Eva, I wanted to give her an idea of, how many patients we have in the freezer and how many do we expect to have. And I just basically emailed all the people I knew and said, please send me your numbers, which they graciously did. But how wonderful it would be to have a database we can go right to. And I think we're going to need to be that super organized to get us through all these regulatory processes. We need to have on record the adverse events that happen, and so those should be obtainable through IRBs, and these would be from the surgical procedures and perhaps things that happen during the cryopreservation process. And also additional safety assessments, as Monica mentioned. Maybe we're going to have to do some more research and say, okay, we will xenotransplant this tissue to show there's no infections occurring. It will be difficult to show efficacy with a xenotransplant, but again, perhaps we can use some of the tissue that's been allocated to research. This will also take time and money that people don't probably have in their grant budgets right now. But also Eva said that they expect the FDA will only need hundreds of cases, not thousands. So that's a good news there. We have to find out for sure. We don't know. And we're not going to have really any efficacy data for the pediatric population anytime soon. So these are the numbers that were graciously sent to me by our colleagues here. And from some of the different centers, the number of cases they had in 2019 and 2020, in 2021, and what they project moving to the future. These are going to be inaccurate numbers, but it's what we had to go on for this initial discussion. So for ovarian tissue cryopreservation, there's about 300, and projected just in the next year, about 200 more. Kyle's group reported yesterday they have 535 testicular tissue samples collected, and this might be an overrepresentation of the data I have in the tables because Kyle's group actually cryopreserves tissues for a couple of these centers that are listed here. And again, another couple hundred projected for the next year. So obviously, this is a growing field as we've been hearing. So what about media for testicular tissue or for ovarian tissue vitrification? Well, Cooper Surgical told us in black and white they will not pursue developing a new media. They'll take care of what we have now, but they're not interested in developing new medias. The main comment I've received after talking to a number of companies is that this is a very small market. Even if you tell them it's growing and it's going to be global and there's going to be thousands of cases right now, this is tiny compared to the other volume of media that is used. Can an already approved media be used? This will require additional testing, and how do we decide what media that should be used? So in trying to figure out what to do about ovarian tissue vit media, I've talked to 4 companies who've all said no, and then I found a wonderful VP of engineering at Cook Medical in their cell and regenerative medicine department. I've had 3 meetings with him so far, and I'm not going to get into all the details that we talked about other than there are places available to make media, not very many of them. He has a very benevolent heart and was very, very interested in this application, and so he was committed to help moving forward. So at the same time, we can develop what's needed for testicular tissue cryopreservation, as well as if we want to pursue vitrification for ovarian tissue. He can do GMP and GLP work, and the actual media production will have to take place outside the US, and he's found 3 places that he currently works with that would be able to do this. So all is not lost, and as David said, we should really do one thing at a time. So Cook also has an excellent regulatory team, just like Cooper Surgical, and he was going to bring all of this to his regulatory team, but we had to cancel the meeting because they felt it was too premature. Until you have a manufacturer of record or a company that says, yes, I'll make this media, then the regulatory teams will talk to you. So it's kind of a catch-22 situation that we're in now. And if we need to go outside for regulatory advice, since we're not a company making media, these meetings can run from $50,000 to $100,000. So we're going to need to find some funding to make this happen. And the next interesting announcement is that Cooper Surgical announced the intent to purchase Cook. So it's not a done deal yet, but this might all be one big happy media family here in the future, which may be good for us in the end. So before we all freak out, what can we do now? So we're still waiting to hear from Eva. We hope to have information to pass on to you before this meeting, but she was not able to get that information from her regulatory team. And she said that their regulatory team is speaking to an external agency to figure out the best path to proceed forward with our requests. Data. These patient registries are just going to underlie everything we do from here on out. And I'm just listing here the 2 that were mentioned at this meeting. The one Maggie Dwiggins and Casey Jones are conducting through Norton Healthcare. It's a REDCap database, voluntary for you to put your data there. They only have 6 centers and 50 patients represented. We know we have a lot more than that. Please put your data somewhere, even now, because we need to get these numbers to Cooper and to the FDA. And we heard the beautiful presentation by Angela Faut and Leslie Apaya's group moving Epic Records to REDCap to capture all these data. and how these two are going to interact. I'm not sure. They can discuss that with you all, but we need to have a uniform database and registry. We just need to also be ready to give Ava any day now when she comes back, any data on adverse events that some of you may have noticed. Hopefully there won't be very many, in addition to the number of cases wherein you have cryopreserved tissue in your liquid nitrogen tanks. And again, to be ready to do additional safety assessment that we might need to get our media approved. So I want all of you to send Eva your positive thoughts, prayers, energy, support, however you do that, so we can get through this initial hurdle of the OC slow freezing medium. And then while we're going through all these difficult processes, to always remember who we're doing this for. Thank you.
Thank you both so far for this information. I just wondered, when we are determining if we want to make the media standard versus humanitarian, perhaps we can involve some IRB individuals from institutions, because with the pediatric population, if we use a specific term that causes alarm, then they will not let us do this in children. So I just want to be sure. Standard works best for IRBs,
Yes.
But obviously I know there are limitations.
Right, and I think that will allow us to grow and be more encompassing as well. Thank you, Leslie.
Yeah, I just want to thank you guys for this very, very important discussion. And I think, from a PIN perspective, this is just so important. And I think traditionally, people come and they're really eager and spending all this time trying to get their OTC protocols open at their institutions and struggling through, does it need an IRB, does it not need an IRB? I mean, it's so burdensome to even get it done. But I think this is like a bit of the elephant in the room of saying we need to make sure we're capturing this information and being really keyed into it, and all those quality control measures. I don't know how much is known, and if there's not a whole lot known about this in this room, I can only imagine what's not known outside of this room. So I just, I hope as we're developing these databases and registries, we're building them to capture what we need, because for these prepubertal patients, we're going to need to be following them for a long time. We're going to need permissions to do that. Mm-hmm. So I don't really have a question, but just kind of a lot of thoughts going through my mind.
Yes,
so.
Thank you. And we'll continue these conversations, and if you want to be in on them, I'm not the boss of this. I just kind of slid in the back door here as my conversations about VIP Media. Please, we need your input. Thanks. Kyle.
Mary, I know that this is not your usual talk, but thanks for giving it. It's, I think, a tremendous service to this consortium and gives us a lot of things to think about. And I would guess that for most of us in the room, we didn't know all of the things that you presented today. So that's a tremendous amount of work for you putting them forward, and David, for you stating so clearly the problem. We do have a risk, though. Cooper has clearly indicated that they're committed to making the media, you say, for 10 months period of time, and that seems like a long period of time. But it's not long if we have to do additional work. I think there's really good news. We probably already do have the hundreds of samples in our liquid nitrogen tanks, including samples that are dedicated to research. But we need to find out really soon what is it that FDA and the Cooper is going to be looking for, so that we can get busy, because 10 months will be over before we're ready to provide the data.
Yep.
I have
as soon as we get back, we'll contact Eva and see where she is. Yeah. Mm-hmm.
I have a feeling that for those of us, and that's probably most of us who are doing this under an IRB protocol, that the adverse event part will be the easy part, because we should be actively accumulating those data as we're recruiting patients. But if there are additional research, well, we need to know soon.
Mm-hmm. Thank you.
Could I have one comment?
Sure.
Sorry.
You go ahead.
Yeah,
then,
sorry.
David.
Excellent talk. I think when you mention the need for a database, one of the things that's coming to mind as multiple are starting, the need to get together and really create a common data dictionary that we can use. And I feel like it's really important to start that work soon. And then there's other models in which we can build from for other studies that are prospectively registering patients to follow along outcomes. So variants of unknown significance in cancer, there's I think, a potential way, rather than everyone getting this through their IRBs, which we know can take forever, would be to have a single-site IRB model where patients can register themselves and then give permission to share data. There's great work out of Penn and Dana-Farber and MSK that we could build upon.
Good idea, thank you. David, and then Veronica.
I'm actually going to answer that.
Oh, okay, go ahead.
because-
Never
So
mind.
just the people who were at the Penn meeting know that we are creating a steering committee for these two databases. We're going to do it together. Leslie used our data collection sheets that we used for the OTC. So hopefully there will be ongoing work, and we'll include you too, so that we can somehow make this better. I mean, we're very aware that both of these databases have issues, and you know, it's a call to arms to get us going. So I'll have more updates for you guys next year.
Thank you, and we all owe both of these groups our deepest thanks for just getting this off the ground. It's not an easy task, so when you see Leslie and Veronica and all their colleagues, say thank you. David.
Yeah, just a couple of comments real quick, mostly keying off of what Kyle suggested, or brought up anyway, is that if you don't have an IRB, you may want to think about that. If you do have one that was deactivated because of ASRM stance, you may want to think about reactivating it just to be safe. It certainly won't hurt to have it. It may not be needed, but I don't think want to wait to the last minute to find out you do need one and don't have it, because it'll take some time. The other thing about producing the medium for 10 months, it does have a shelf life, typically about a year. So there may be some time after that, but still, it's a finite period. So.
Thank you. And I just want to add about the IRB. You might want to reach out to those of us who have an IRB so that you're not recreating the wheel, and take several and see how your institution wants it to look. It can be an IRB where you are just following outcomes. You don't need to have a research question, which I think is the kind of trick when you're speaking with your IRB. They'll say, well, what is a research question? So you need to be sure that you're saying we're just following outcomes. So several of us have IRB protocols that we're willing to share, so you don't have to recreate the 2-year wheel that will take.
Thank you, Leslie.
Yeah, I was just going to speak to that exact point, because the whole, deactivating of the IRB and reactivating of it, the reason that's necessary is because it's in a different context now, right? So before, it was the IRB was there because under the entire semblance of the ASRM saying this was experimental, which then that changed. So it does force a change that needs to happen to the IRB, but I agree, an IRB is still really important to follow these outcomes and to answer these questions.
Great, thank you all for your comments. And when we ask you for data coming up here quick, please respond. Thanks.